Induced pluripotent stem cells. Induced pluripotent stem cells are a type of pluripotent cell that is produced from unipotent stem cells. The unipotent cell may be almost any body cell. These body cells are then generally altered in a lab to make them acquire the characteristics of embryonic stem cells, which are a type of pluripotent cell. To make the unipotent cell acquire the new characteristics involves inducing genes and transcriptional factors within the cell to express themselves. In other words, to turn on genes that were otherwise turned off. The fact that these genes are capable of being reactivated shows that the adult cells retain the same genetic information that was present in the embryo. The iPS cells are very similar to embryonic stem cells in form and function. However, although they express some of the same genes that are usually expressed in the embryonic stem cells, they are not exact duplicates of them. One feature of particular interest is that they are capable of self-renewal. This means they can potentially divide indefinitely to provide a limitless supply. As such, they could replace embryonic stem cells in medical research and treatment, and so overcome many of the ethical issues surrounding the use of embryonic stem cells in research. Pluripotent cells in treating human disorders. There are many possible uses of pluripotent cells. The cells can be used to regrow tissues that have been damaged in some way, either by accident, for example skin grafts for serious burn damage, or as a result of disease, e.g. neurodegenerative diseases such as Parkinson's disease. Some potential uses of human cells produced from stem cells. Heart muscle cells. These can be used for heart damage, for example as a result of a heart attack. Skeletal muscle cells can be used in the treatment of muscular dystrophy. Beta cells of the pancreas can be used to treat type 1 diabetes. Nerve cells can be used to treat Parkinson's disease, multiple sclerosis, strokes, Alzheimer's disease, and paralysis due to spinal cord injury. Blood cells can be used to treat leukemia and inherited blood diseases. Skin cells can be used to treat burns and wounds. Bone cells used to treat osteoporosis. Cartilage cells used to treat osteoarthritis. And retina cells of the eye could be used to treat macular degeneration. Human embryonic stem cells and the treatment of disease. Although there are a number of types of stem cells in the human body, it is the first few cells from the division of the fertilized egg that have the greatest potential to treat human disease. As they come from early stage of the embryo, they are called human embryonic stem cells. These cells can be grown in vitro and then induced to develop into a wide range of different human cells and tissues. At present, embryonic stem cell research is only allowed in the UK under licensed and specific conditions. These conditions include its use as a means of increasing knowledge about embryo development and serious diseases, including their treatment. Embryos used in this type of research are obtained from in vitro fertilization. The process nevertheless presents a number of ethical issues. One issue surrounds the argument as to whether a human embryo less than 14 days old should be afforded the same respect as a fetus of an adult person. Some people feel that using embryos in this way undermines our respect for human life and could progress to the use of fetuses and even newborn babies for research or the treatment of disease. They feel that it is further move towards reproductive cloning and, even if this remains illegal in the UK, the information gained could be used to clone humans elsewhere. Others disagree, arguing that an embryo at such an early stage of development is just a ball of identical undifferentiated cells bearing no resemblance to a human being. They feel that the laws prohibiting cloning in the UK and elsewhere provide sufficient protection. Supporters of human embryonic stem cell research contend that it is wrong to allow human suffering to continue when there is a possibility of alleviating it. They further argue that since embryos are produced for other purposes, for example fertility treatments, it makes no sense to destroy spare embryos that could be used to do research. Opponents of embryonic stem cell research contend that it is wrong to use humans, including human embryos, as a means to an end, even if the end is to alleviate human suffering. However, human embryos are not the only source of stem cells. For example, they can be obtained from bone marrow of human adults. As long as a person gives consent, this source of stem cells raises no real ethical issues. At present, these cells have far more restricted medical applications, but scientists hope in time to be able to make them behave more like embryonic stem cells. Growth of plant tissue cultures. Mature plants have many totipotent cells. Under the right conditions, many plant cells can develop into any other cell. For example, if you take a cell from the root of a carrot, place it in a suitable nutrient medium and give it chemical stimuli at the right time, we can develop a complete new carrot plant. There are many factors that influence the growth of plants tissue cultures from totipotent cells. One group of factors consists of plant growth factors, which are chemicals involved in the growth and development of plant tissues. Plant growth factors have a number of features. They have a wide number of effects on the plant tissues. The effect on a particular tissue depends upon the concentration of the growth factor, and the same concentration affects different tissues in different ways. The effect of one growth factor can be modified by the presence of another. An experiment was carried out to investigate the effects on the development of plant tissue cultures of 3 growth factors, cytokinin, indole-acetic acid, and 2,4-D. Samples of totipotent plant cells were grown on a basic growth medium in a series of test tubes. Each test tube contained a mixture of the 3 growth factors in different concentrations. After 2 weeks, the tubes were observed to see the effects of the growth factor on the shoot and the root growth. So, for example, in one tube there was no cytokinin, very low IAA, and no 2,4-D, and the shoot developed moderately and the root developed very little, indicating that IAA, even in low doses, does very little to the roots. It has to be in absolutely minute doses to make the roots develop, and in high doses will make the shoots develop. Whereas with lots of cytokinin, there is some root development and no shoot development, showing that cytokinin is involved in the development of the root. And in contrast, 2,4-D appears to work very well on the shoot development and on the root development.